Hear me okay? It's Tuesday. Good morning. It is 9:01. We're going to get started. Any questions, any issues, any problems? Anything at all, guys? And nobody's going to say anything, but they're still going to ask. Uh, just to remind you, we do have an exam already next week, Thursday, right? You are keeping an eye on the schedule. So we already have our third exam coming up next week, Thursday. So just a little bit over a week until our next exam. Okay. And this week we're going to be covering two chapters. We're going to cover chapter 10 and chapter 11 this week. And then next week, Tuesday, we're going to cover chapter 12. And then we're going to have our third exam. Okay. Keep an eye on that. All right. Let me share my screen with you. And today I want to start and finish chapter 10, and hopefully I can also start a little bit of chapter 11 just to make sure we are able to finish everything within this week. And the chapter 10 is going to be about our blood. So all about the blood. We're going to talk about the functions of blood. Why do we have it? Why do we need it? We're going to talk about the composition of blood. What is in the blood? What are the different cells? Why do we need them? Two very important terms that you guys need to understand and differentiate. One is what they call hematopoiesis. The other one is called hemostasis. Make sure you don't confuse the two. Hematopoiesis is the process of new blood cell formation, okay, which is going to happen inside the red bone marrow, and it starts with a stem cell called the hemocytoblast. Hemostasis is the stop the flowing of blood, is the clotting of blood, which is a very complex process. And we're going to talk a little bit about it today. Okay. And then we're going to finish this class talking about blood group. The folks that are in my lab on Monday that we just had yesterday, you have a legs up because we already started the discussion of blood in yesterday's slide. So why do we need the blood in our body? Number one, we need the blood in order to obtain our oxygen gas, O2, right? And we know that oxygen gas is going to bind to the red blood cells, to the hemoglobin in the red blood cells. And then as the red blood cells in our blood circulate all over the body, they are going to distribute the oxygen gas. Now, that is not the only reason why we need the blood. We also need to discard the CO2. All the CO2 that is made by every single cell in the body during the process of aerobic respiration, we need to get rid of it. CO2 is toxic. We don't want the CO2. And the CO2 is going to go in our blood, it's going to diffuse in the blood. And then when it gets to the lungs, now we are going to send the CO2 into the alveoli so that we can exhale the CO2 and get rid of it. Now, the blood is also where we're going to transport nutrients. So the stuff that we eat eventually gets into our small intestine and is going to be absorbed. The nutrients are going to be absorbed by the blood, and then it's going to be distributed all over the place. So blood is very important for that as well. And also we have messengers in our blood. What kind of messengers, guys? We have hormones, right? The hormones, they're going to be distributed and circulating all over the world. Wei, you had a question?
Sorry, Professor. I'm trying to download the PowerPoint. I don't know why it says the file has been deleted.
Oh,
it's not found.
okay. I'll take a look at that after class to make sure that it's available. Okay. Can't do that. Now let's take some closer look at the functions of the blood. And blood is extremely important to maintain our homeostasis. And by now, we all know what homeostasis means. We know that homeostasis means balance in our body. So to help to maintain this balance, blood plays a huge role on that. Again, blood is going to help the transportation of gases like oxygen gas, CO2, nutrients, and messengers, hormones, and cytokines. Within our blood, we also have our protection system, our immune system. And we're going to talk more about the cells that are involved within the immune system, but that is a huge, huge function of our blood to help to protect our body. And our blood helps with the regulation of our pH, the pH of our body. It helps with the regulation of our body temperature, and it helps with the regulation of blood osmotic pressure, maintaining the right concentration of solutes in the blood to make sure that the cells that are within the blood, they're able to survive and thrive. Okay? We don't want a hypertonic environment in our blood. We don't want a hypotonic environment. Our blood needs to be isotonic. Okay? Now, what are some of the characteristics of blood? Well, we all know, I'm sure that the first thing that comes to your mind when we think of blood, we're going to think of the color red, right? Blood is red. Now, the reason why blood is red is because of the erythrocytes, the red blood cells. We're going to talk more about them in a minute. But this red coloration of blood can differ depending if the blood is rich in oxygen or if the blood is poor in oxygen. Oxygen-rich blood has a bright red coloration to it. So if you cut an artery, and arteries carry oxygen-rich blood, usually in the systemic circulation, you're going to find a very bright red coloration of the blood. Now, oxygen-poor blood, this one is going to have a dark red coloration. So it is different from oxygen-rich, okay? Now, what about the pH of our blood? Our blood, folks, is slightly alkaline. It has a blood pH between 7.35 and 7.45. Again, just above neutral. Okay? It's not acidic, it's not neutral, it's slightly alkaline. And you can see here that the range in the pH of blood is very narrow. Is only 0.1 in the pH scale from 7.35, 7.45. And that is very important to be maintained because if the pH deviates from this right here, this can be very, very dangerous and it can actually kill an individual, okay? Because that is going to cause the destruction of the blood cells and destruction of the proteins within our blood. The volume of blood varies between male and female. In general, females have between 4 to 5 liters of blood, while males, simply because we have usually a bigger body, we have between 5 and 6 liters of blood. And blood is more viscous than water. What does it mean, viscosity? Guys, viscosity is the amount of things, of solutes that are dissolved in a fluid. Right? We have a lot of proteins. We have a lot of ions. We have a lot of cells that are within our blood. And that makes blood more viscous than water. And our blood is slightly hotter than our core body temperature. Blood temperature is around 38 degrees Celsius, and the body core temperature is about 37 degrees Celsius. All right. Now let's take a look at the components of blood. What do we find in our blood? If you ever wonder what happens when they take the blood from you, even because you're donating blood or you're doing a blood test, okay, a blood analysis, they're going to take your blood and they're going to put your blood in a flask, just like this one right here. And then they're going to take this flask and they're going to put this flask at an angle inside of a machine called a centrifuge, right here. And again, you can see how the flasks are placed at an angle, okay? Now, what are they going to do? They're going to turn on the centrifuge, and centrifuges, they spin really, really fast. And what they do is that they're going to separate the components of the blood by weight. Whatever is heavier in the blood will go to the bottom of the tube. Whatever is lighter is going to stay at the top of the tube. Okay, so after we take the flask out of the centrifuge, after we spin around for a while, this is what the flask is going to look like. We're going to see this clear section right here at the top of the flask. This fluid, this clear fluid, is what we call the plasma. Okay, and the plasma is mostly water, is about 90% water. And within the plasma, we're going to find gases dissolved in it. We're going to find proteins. And the plasma makes up about 55% of our blood. So more than half of our blood is basically water. Then at the bottom here, below the clear section, you're going to find what we call the formed elements, which is about 45%. Now, formed elements is just a fancy name for the cells within our blood. Okay? All the cells, because they are heavier, they're going to accumulate at the bottom of the flask. Now, in between, right below the clear layer, the plasma, we're going to find this cream-colored layer right here. This is what we call the Buffy coat. The Buffy coat. And within the Buffy coat, we're going to find our white blood cells and we're going to find our platelets. And we're going to talk more about each one of those cells. And then the rest of the formed elements, we're going to find the red blood cells. So you can clearly see that we have a lot more red blood cells than white blood cells and platelets in our blood. Okay? Any questions? All right. Now, let's take a closer look at the plasma and let's see what we find in the plasma. So the plasma, as I mentioned earlier, is mostly water, about 90% water, and then we have about 10% solutes dissolved within the water. Now, among the solute, what are we going to find? We're going to find the nutrients, glucose, fatty acids, amino acids. We're going to find dissolved gases, CO2, O2, and we're going to find hormones and cytokines, all dissolved within the plasma. We're also going to find salts, sodium, potassium, magnesium, calcium, etc. And we're going to find the different proteins. Among the proteins, we're going to find a protein called albumin. And albumin is a protein that is very important in our blood to help to maintain the osmotic pressure of the blood. Again, what does that mean to maintain the osmotic pressure? To keep our blood isotonic. Okay? So that the cells that are in the blood, they're not going to burst, they're not going to shrivel and die. They're going to be happy within that isotonic. All right, now what about the formed elements? Again, formed elements are the cells. What are the different cells we find in our blood? We're going to find the red blood cells, and the red blood cells are also what we call the erythrocytes. Now, erythro means red, cyte means cell. So these are the red blood cells. And the function of the erythrocytes, the red blood cells, is to carry oxygen and distribute the oxygen all over our body. Okay? Then we have the white blood cells. The white blood cells are also what we call the leukocytes. Leuko means white. And the leukocytes are involved with our immune system. They help to protect our body against anything that is foreigner to our body. And we're going to talk about some of these leukocytes. Then we have the platelets. And the platelets are also called the thrombocytes. Thrombo means clot. Okay? And the platelets, the thrombocytes, they are involved with the formation of clotting. So to make sure that we don't bleed to death. So we're going to form the blood clotting when we cut a blood vessel. Right? Now, let's say that I take a sample of blood from a healthy individual. And I take the drop of blood and I put it into a glass slide and I look under the microscope. This is what I would see, folks. What are we looking at right here? We're looking at a drop of blood from a healthy individual under the microscope. And we can see all these red blood cells. So we can see how many we have there. They're much, much higher number than any other cell in the blood. We can see the white blood cells, which are these bigger cells right here. And then we can see these smaller little fragments of cells, and these are the platelets. Okay, and they're all floating in what? They're all floating in our plasma. Okay, so the ratio of red blood cell per white blood cell and platelet is 1,000 red blood cell for 1 white blood cell and 40 platelets. Okay, again, red blood cells are much, much more abundant. Two important terms that you guys need to know that I do like to ask questions about it in the exam. One is what we call the hematocrit. The other one is anemia. What is a hematocrit? A hematocrit is a type of blood test that we can do. And this blood test is going to check for the percentage of red blood cell within our blood. Okay? For females, the percentage of red blood cell is between 38 and 46%, while for males, the percentage of red blood cells is about 40 to 54%, okay, of our entire blood. Now, what if you do a hematocrit test and then you get the result from the hospital and your results show a lower percentage of red blood cell than the average, than the normal right here? So guys, now you are officially anemic, okay? Because anemia is a decrease in the number of red blood cells. Now, there are several things that can cause anemia, and we're going to go over some of them, but anemia simply means a drop in the number of red blood cell below the homeostatic level, below the average. Okay, you don't know what is causing it, but that's what anemia is. And we're going to talk more about it in a little bit. Now, let's go over each one of the formed elements in a little bit more detail, starting with the erythrocytes, the red blood cells. Guys, red blood cells are very unique cells in our body. Okay, there's nothing else that looks like the red blood cells, that perform like red blood cells. First of all, red blood cells, they have a biconcave shape to them. You know, imagine if I took one of the cells right here and I split it in half like this, okay? And I'm looking at the side view, this is what it would look like. Kind of look like a donut, but it doesn't have a hole in the middle. It just have an indentation right there in the middle. It has a biconcave shape. Now, does a red blood cell has a nucleus? No. Mature red blood cells are anucleated. They don't have a nucleus. Does a red blood cell going to have a protein machinery, like ribosomes, for example, to make proteins? Of course not. If you don't have a nucleus, you don't have DNA. If you don't have DNA, why would you have protein machinery to make proteins? Right? You can't make protein from nothing. And the reason why red blood cells, they lost all of these structures, all of these organelles, guys, during the process of evolution, is because red blood cells became extremely specialized in what it does. What is that they do? They carry oxygen gas, okay? And to carry oxygen gas, they're going to have a protein called hemoglobin. So the red blood cell got rid of all the organelles inside, the nucleus, the all these different organelles that we have, ribosomes, centrosomes, etc., so that we can fit in more hemoglobin on the inside. Now you might be wondering here, wait a second. I always watch the CSI show and those detective shows, and they collect DNA from a blood sample. And you're telling me that there is no DNA in red blood cell? Yes, I'm telling you that. So where do they get the DNA from in blood? From the other cells. We don't have just red blood cell in blood. We also have white blood cells and we have the platelets as well. This is where the DNA comes from a blood sample, not from the red blood cells. Okay? And because the red blood cells, they lost all of these organelles, they don't survive very long. The half-life of a red blood cell is only about 120 days at most, and then they're going to die. Okay? So what is left inside the red blood cell? This guy right here, folks. Lots and lots and lots of hemoglobin. What is a hemoglobin? A hemoglobin is a protein that is responsible for the attachment and distribution of oxygen gas in our body. Hemoglobins are very interesting. Hemoglobins are made of actually 4 different protein units. They're going to combine together to form this bigger protein called the hemoglobin. Okay? Now we can see that in the center of each one of these protein units, the polypeptides, and we have 2 alpha chains and 2 beta chains. In the center, we are going to find this weird looking structure right here, kind of looking like a flying saucer, right? Right there. What is this? That is a heme, hence the name hemoglobin. Globin means protein. Heme comes from this center section of the hemoglobin right here. Now, if I take the heme and I draw a chemical structure of the heme, this is what the heme would look like, okay? Now, I want you guys to focus on the center of this chemical structure, right there. We are going to find an iron core in the middle of the heme. Why do we have a metal? Why do we have iron in the middle of the hemoglobin? Guys, the purpose of the iron is that iron is a oxygen scavenger. Iron can find oxygen gas and bind to the oxygen gas. And once the oxygen gas is bound to the iron in the hemoglobin, now as the red blood cells circulate all over the body, they're going to distribute the oxygen gas to every part of our body. But if you don't have the iron right here, folks, then you can't transport oxygen gas, O2. In fact, your red blood cells are going to collapse. They're going to die. And that's going to lead to anemia. This is what we call iron deficiency anemia. Okay? Now, one of the problems, though, with the heme in the hemoglobin is that the heme doesn't bind just to oxygen gas. The heme in the hemoglobin also binds very strongly with carbon monoxide, CO. And folks, when we are exposed to carbon monoxide, okay? By the way, how do we get carbon monoxide? When you burn fuel, if you burn a wood log, if you burn gas, running your car, all of these things, they release carbon monoxide. And if you are exposed to carbon monoxide at a high concentration in a closed environment, carbon monoxide is going to bind to the heme in the hemoglobin, and that is going to prevent the binding of oxygen gas. What's going to happen to that individual? That individual is going to die. If you don't find that individual right away and you remove from that place and give 100% oxygen gas to breathe in, to displace the carbon monoxide, that individual is going to die within 5 minutes. Being exposed to carbon monoxide is extremely, extremely dangerous. Any questions, folks? All right. Now, let's take a look at one disease that is associated with our red blood cells. And this disease is a condition called sickle cell disease. First of all, this is a genetic condition. It's something that you are born with. Number two, I will ask you questions about this in our next exam next week, Thursday. How more clear do I need to be? You tell me. Just tell me what do you want me to do to make sure that you get this question right in this. Okay? So you need to know the things we're going to be discussing right now. If something that I say about sickle cell disease is not clear, please stop me and ask questions. Okay? Now, on the left-hand side, we can see a normal, happy red blood cell. If you don't believe me, just look at it. It looks happy. And we can see that this red blood cell here has this sequence of amino acids. And the sequence of amino acids make a normal beta chain of the hemoglobin. Remember, the hemoglobin is made of 2 alpha chains and 2 beta chains, right? And what we're looking at right here, we're looking at a little sequence, amino acid sequence of the beta chain that makes up the hemoglobin in a normal red blood cell. So we have valine, histidine, leucine, tryptophan, proline, glutamine, and another glutamine. Okay? On the right-hand side, we have a sickle cell red blood cell. You can see the shape is completely different. It is a flat-looking cell and looks very unhappy. Now, what's going on with the sickle cell? How does it differ from a normal red blood cell? Right here, folks. We can see right there at that amino acid position, instead of having a glutamine right here, we are going to have a valine. This is what we call one amino acid substitution. And because of this wrong amino acid right there, the entire hemoglobin will collapse. The hemoglobin cannot maintain its shape. Because remember, the shape of a protein is very important for this function. So if the hemoglobin falls apart, the entire function of the red blood cell is going to suffer as well. Okay? So sickle cell is a genetic disorder in which there is a single amino acid substitution in which we're substituting a valine for a glutamine, and that causes the collapse of the red blood cell. Okay? And this faulty hemoglobin chain, so this hemoglobin beta chain right here, this is going to cause the cells, the sickle cells right here, to clump together. Because of their shape, they're going to start clumping with one another. They're not going to flow like we see usually in a normal, healthy red blood cell through the blood vessels. They start clumping. Okay? And because of this sickle cell shape here, these cells become very fragile. So what are some of the complications associated with the sickle cell disease? Number one, we're going to have the clogging of the blood vessels because they are clumping together. Number two, they rupture very easy because they are very fragile. And that is going to lead to what? That is going to lead to anemia. The type of anemia that we call sickle cell anemia. Okay? Now, sickle cell disease is a type of genetic disorder that is called autosomal recessive disorder. I don't want to lose you. I don't want to overcomplicate things, but I do want to explain what this autosomal recessive disorder means. It's actually quite simple, and I need everybody's attention here. Okay, folks, we know that every gene in our body, we have two copies of that gene. Why? One copy coming from the sperm, from your dad, the other copy coming from the egg, from your mom. So every gene in our body, we have two copies of it. Okay? In an autosomal recessive disorder, what this means is that in order to have the sickness, in order to have the disease, both copies of your beta chain gene will be mutated. So both copies that you have that makes the beta chain for the hemoglobin, they will be mutated. They're going to have this mutation right here, the single amino acid substitution, and therefore that leads to the disease. If you only have one copy mutated, but the other copy is normal, you will not have the disease. You're going to have you're going to be a carrier of the disease, of the mutation, but you're not going to have the disease itself. So you're going to have the genotype, but you're not going to have the phenotype. Okay, again, you need both copies mutated in order to have sickle cell disease. That's what autosomal recessive means. Good? Any questions, folks? Anything at all from this slide right here, sickle cell disease? It's not. Now, sickle cell, again, it is a genetic disorder, is not something that is very common in the general population, but it is something that is more prevalent in African Americans, and it is very common in the African continent, especially in countries in Africa where we have a high incidence of a condition, a disease called malaria. Okay? What is malaria, folks? Malaria is a infectious disease caused by a parasite and which is distributed by the bite of mosquito bite. If a mosquito is carrying the parasite and then the mosquito bites you, then it's going to drop the parasite into your blood, and the parasite likes to survive, likes to live and propagate inside red blood cells. Inside the red blood cells. Now, if an individual has a sickle cell red blood cell, the parasite cannot survive inside a sickle cell red blood cell. They like normal red blood cells. Okay? So what happens is that in Africa, there was a selective pressure in the population to keep at least one copy of the sickle cell gene alive in the population. Why? Because if you have one copy of the gene mutated, but the other copy is normal, you don't have sickle cell disease, but you are protected against malaria. And because of that protection against malaria, you are going to be able to survive until adulthood, until your reproductive age. And then you're going to mate, you're going to find a partner, you're going to have kids, and you're going to pass on your gene to the next generation. So in Africa, in places where, again, there's a high incidence of malaria, it is better for you to have one copy of the gene mutated and the other copy normal than it is to have both copy normal or both copies mutated. I mean, if both copies are mutated, you have sickle cell disease, and that's very bad. If you have both copies normal, you're going to develop malaria, and that could be bad. It could be fatal. But if you have one copy mutated and the other copy normal, that you're not going to have sickle cell, and on top of that, you're going to be protected against malaria. Okay? So we're gonna watch. I think I've removed the video, but there's a video that I'm gonna post later that talks a little bit more about this evolutionary pressure in which the sickle cell gene remained in the African population.
I'm fast, Professor. Sorry, I had a question. Is there any treatment or anything for individuals that do have sickle cell, or is it just a always fatal disease?
Question: if you have a mutation of both of the genes, it is fatal. It is fatal. It's very painful because you get all these cloggings of your blood vessels, and there's really no treatment. Yeah. But if you only have one copy of the gene mutated and the other one normal, then you're gonna be you might have here and there some signs and symptoms, but overall you're gonna be just fine. Any other questions? All right. Now we have determined that anemia is a decrease in the number of red blood cells, right? We established it. But like I said before, several things can lead to anemia. One of them is sickle cell, right? Because if you have a sickle cell and your red blood cells are very fragile and they're going to die very easily, that is going to decrease the number of red blood cells. We also have what is called iron deficiency anemia. Like I mentioned earlier, if you don't have enough iron, your hemoglobin is not going to be built, your red blood cells are going to die and collapse. So that's going to lead to anemia. And we also have a type of anemia called pernicious anemia. And pernicious anemia is related with a lack or deficiency in vitamin B12. And also a lack of a protein called intrinsic factor. What is the job of B12 and intrinsic factor? Guys, B12, the vitamin B12, is essential for hematopoiesis. Okay? So if you don't have enough vitamin B12 in your red bone marrow, hematopoiesis is going to suffer. And if you don't have enough hematopoiesis, your number of red blood cells are going to decrease. Now, in order for vitamin B12 to be absorbed into our cells, we need a protein that is made by our stomach. This protein is called intrinsic factor. And some people, some folks, they might have a deficiency, a genetic problem, a genetic deficiency, in which the amount of intrinsic factors that is produced by the stomach is low. So if you have low intrinsic factor, even though you might have enough vitamin B12, the vitamin B12 will not be absorbed into your cells. Just like vitamin D and calcium, right? In order for the vitamin calcium to be absorbed by the cells, we need the vitamin D. In order for the vitamin B12 to be absorbed by the cells, we need intrinsic factor. And if you don't have enough, that's going to lead to a type of anemia called pernicious anemia. All right, moving on, let's talk about the white blood cells now, also called the leukocytes. Folks, unlike the red blood cells, white blood cells are like normal cells that we have talked about before in class. They have a nucleus. They have a ribosome. They have all the different organelles that we usually find in us. Okay? Now, we have seen that we have a lot less white blood cells than red blood cells. And one thing that is special about white blood cells, one unique characteristic of white blood cell is that they can do something called diapedesis. What is diapedesis? Diapedesis is the capacity of white blood cells to migrate in and out of the blood vessel. They can literally squeeze themselves in between the walls of the blood vessel so that they can go to the site of infection. So let's say we have a lot of microbes right here, bacteria. You cut yourself with a dirty knife. Okay? We want the white blood cells to get out of the blood vessel, go to where the bacteria is, and start breaking them down, destroying them. And that's what we call diapetesis. Okay? The major function of white blood cells is immune response against anything that is foreign to our body. And we do have two major types of white blood cells. And we're going to talk more about them. What are those two major types? We have white blood cells that contain granules. We call them the granulocytes. And we have white blood cells that do not have granules. We call them agranulocytes. So these are the two main types of white blood cell, the granulocytes and the agranulocytes. Let's see what are the white blood cells that belong to the granulocyte family. And let's see the white blood cells that belong to the agranulocytes. Okay? Within the granulocytes, we have neutrophils, eosinophils, and basophils. Let's talk a little bit about each one of them. Neutrophils are the most abundant white blood cells in our blood. Most abundant. And the function of the neutrophils is that they are phagocytic cells. Remember what phagocyte is? Phagocytes, they engulf other cells, they take them inside and destroy them. That's what the neutrophils do. The eosinophils, they're involved with killing big parasitic worms, like the malaria parasite, for example. And they don't kill by doing phagocytosis. They kill by releasing cytotoxic enzymes that is going to destroy the large parasites. Then third, we have the basophils. And the basophils are involved with allergic reaction. And they are the one that release histamine when you have an allergic reaction. Guys, if you have allergy to anything, these are the guys to blame right here. These are the cells to blame. Okay? Because when you have an allergic reaction to something, this means your immune system is hyperactivated. And in the process of activating the immune system, the basophils, they send a chemical out called histamine. Histamine, the function of histamine is to alert the immune system of something wrong in our body. But in the process of doing this, activating the inflammatory response, histamine causes all the symptoms that we have when we have an allergy. The accumulation and extra production of fluid, redness of the eyes or the skin, fever, all those things are caused by the release of histamine. Folks, what is an over-the-counter medication that we can take when you're having an allergic reaction? I'm not asking about the specific name of the drug. I'm talking about the type of drug. What can we take is an over-the-counter when you have come on,
The
future help.
antihistamine.
Antihistamine, absolutely. An antihistamine. What does it do? It prevents the release of histamine by the basophils. Okay? Now, among the agranulocytes, we have the lymphocytes. And the lymphocytes, we have the T cells and the B cells, and they're involved with killing of microorganisms. And the B cells are involved with secretion of antibody. We're going to talk more about that later. And then we have the monocytes. And the monocytes, just like the neutrophils, they're also phagocytic cells. Okay? They can do phagocytosis. Any questions? All right. Another two terms that I want you guys to know and be able to differentiate. One is what we call leukocytosis. The other one is called leukopenia. Cytosis, guys, is an increase in white blood cells. Can anybody give me an example of something happening to our body that will lead to leukocytosis?
Infection?
Yes, an infection. Every time that you're fighting an infection, every time that you have a cold, every time that you cut yourself and you have microbes inside of your body, that is going to lead to an increase in the white blood cell so that they can fight that infection. Okay. Now, leukopenia is the opposite. Leukopenia is a decrease in the number of white blood cells. And some cancers can cause leukopenia. It can cause a decrease in white blood cells. Okay? Now, since we are talking about white blood cells and immune system and cancer, let's talk about one type of cancer called leukemia, which is a unique type of cancer because leukemia is not a solid tumor. Leukemia is a cancer of the red bone marrow, in which the cells of the red bone marrow, they become defective. Okay? Now, there are different types of leukemia depending on the type of white blood cell that is affected. If you have, for example, lymphocytic leukemia, like what we're looking at here on the screen, so lymphocytic leukemia is leukemia of what kind of cells, guys? The lymphocytes. The lymphocytes are defective. Okay? Now, in leukemia, we're going to have a massive increase of white blood cells. But wait a second, that sounds counterintuitive because most of us know that someone that has leukemia, their immune system is very weak. Right? A simple cold can kill an individual with leukemia. Now I'm telling you that there is this massive increase in white blood cell. What's going on here? This is what is going on, folks. The problem is that even though there is a massive increase in white blood cells, someone that has leukemia, the white blood cells are immature. And because they are not mature, they are not ready to provide immune function. They don't work as they should. Yes, we have all of these white blood cells, but they are all non-functional. And now the patient is going to become very, very prone to infection. Take a look over here, folks. I don't know if you remember, Phil, at the beginning of class today, when we look at a sample of blood from a healthy individual, and we have all these red blood cells, and then a few white blood cells and a few platelets. Look at this, guys. These right here are the red blood cells, okay? And then all of these are all immature white blood cells. Look how many we have right here. You can clearly see just by looking at this blood sample that this individual is having leukemia due to this massive increase of immature white blood cells. Any questions? Let's talk about the platelets now. And the platelets are kind of interesting because they are not actually full cells. Platelets are fragments of cells. And a platelet is actually formed from a cell called a megakaryocyte. Mega means big. So we have this big cell right here. And then this big cell will basically disintegrate. It will fall apart into smaller fragments. So a platelet doesn't have a full nucleus on the inside. It has the little fragments of the nucleus, but it doesn't have the full nucleus. Okay? And because of that, again, they are very short-lived. They only survive for about 5 to 9 days total, but they do have a very important function. They help to clot our blood. There's a condition that is related with the thrombocytes and some other clotting factors, and this is a condition called hemophilia. also called the bleeder's disease. Why is it called the bleeder's disease? Because this individual here cannot clot his blood or her. Okay, now, where does all the blood cells come from? We already know that. It does come from inside our red bone marrow. And the process of formation of new blood cells is what we call hematopoiesis. Now, pay attention. I'm not just saying red blood cell formation, okay? I'm talking about all blood cell formation, right? And the entire process starts with one type of cell called a hemocytoblast. And this hemocytoblast here, folks, this is a little blood stem cell. Remember, we have talked about stem cells before. A stem cell is an undifferentiated cell that can transform into other cells. So we have stem cells in our red bone marrow. And the stem cell here, depending on the messenger, on the signal that it receives, and the signal are hormones, different hormones, the hemocytoblast can differentiate into the lymphoid stem cell or the myeloid stem cell. The lymphoid stem cell will become the lymphocytes, T cells, and the B cells. The myeloid stem cells are going to become the other blood cells, the erythrocytes, platelets, and the different type of white blood cell, macrophages, monocytes, eosinophil, basophil, etc. Okay? Now, as you can imagine, hematopoiesis needs to be tightly regulated. It is such an important function in our body to make the blood cells. It's hugely important. So the process will be tightly regulated. And what is going to regulate hematopoiesis, folks? Hormones. Hormones, different type of hormones will regulate formation of different blood cells. Let's see what are the hormones regulating different blood cells. Let's start with the red blood cells. For the red blood cells, we're going to have a hormone called erythropoietin. And erythropoietin is actually made by our kidneys. Right here. It's not even a major endocrine organ. But the kidneys, just like many organs in the body, they do make different hormones. One of those hormones produced by the kidneys is erythropoietin. And erythropoietin is going to go all the way inside the red bone marrow and is going to activate the hemocytoblast to differentiate into a myeloid stem cell, which will then differentiate into a red blood cell, increasing the number of red blood cells in our blood. What about for the platelets? We have a different hormone called thrombopoietin, which is also produced by the kidneys. So again, when the platelet number is low, the kidneys are going to secrete thrombopoietin. Thrombopoietin, the hormone, is going to go to the red bone marrow and is going to lead to the differentiation of the hemocytoblast into platelets. And finally, for the white blood cells, there are two different types of hormones. We have a colony stimulating factor, the CSF, and another hormone called interleukin, okay? Which then is going to produce the different types of white blood cells. I do like to ask questions about this in these FYI. Now, we have just established that red blood cells don't live long. They only have about 120 days at most. So what's going to happen when a red blood cell is old and worn out? Are we just going to take it and dump it in the trash like we do with everything else at home? No. Remember, our body is much more conscious than we are, thank God. So our body, they're going to take these red blood cells and is going to recycle them. When the red blood cell gets all the way inside of the spleen, folks, this is one of the functions of the spleen, in fact. In the spleen, we have macrophages that will take the red blood cells and do phagocytosis of the old, worn out red blood cells. We're going to break them down. Once they are broken up, the body is going to take the globin, which are the proteins. Globin means peptide, proteins. They're going to take the globin and they're going to recycle it. So it's going to reuse all the different amino acids from the hemoglobin. Okay? And they're going to take the iron and they are going to recycle that as well. So they're going to take up and recycle some of that iron, and that's going to be done by the liver. Now, the rest of the heme, besides the iron, the rest of the heme cannot be recycled by the body. So the body is going to take the heme and is going to transform it into a pigment called bilirubin. Then this bilirubin is going to go to the liver, and the liver is going to get rid of the bilirubin. How? It's going to get rid of the bilirubin through our feces and get rid of the bilirubin through our urine. Folks, the reason why our urine is yellow is because of the presence of urobilin. Okay? And the reason why our stools, our poop is a little yellowish, brownish, is because of a bilirubin, a pigment from the bilirubin called the stercobilinogen. Right? And so we get rid of the bilirubin. Now, sometimes the liver is not working properly. The liver is malfunctioning, and instead of getting rid of the bilirubin, we're going to have accumulation of bilirubin in the liver, and now this bilirubin is going to go into the bloodstream, and that is going to cause a condition called jaundice. So what is jaundice? Jaundice is the yellowish of the skin and the eyes due to the accumulation of bilirubin in the blood because of damage to the kidneys. The kidneys are not draining properly, the bilirubin. So we talked about hematopoiesis, and now let's talk about hemostasis. And again, what is hemostasis? Hemostasis is the process to stop blood flow, okay, to reduce blood loss if you cut yourself. Now, I do like to ask questions about this in the exam, FYI. Let's see what is the process of hemostasis. And I mentioned earlier, is actually a very complex process. And I'm going to explain to you why it needs to be complex, just a little bit, okay? Now, let's say you are cooking in the kitchen, and you take a knife and you accidentally slice your finger. Immediately, you're going to start bleeding, right? As soon as you start bleeding and you damage the blood vessel, we're going to have something called vascular spasm. What is vascular spasm? Vascular spasm is the constriction of the blood vessel. In order to what, folks? In order to decrease the amount of blood loss. Remember, on the walls of the blood vessels, we have smooth muscle. And the smooth muscle is going to contract, causing the constriction of the blood vessel, decreasing the amount of blood loss. Next, we're going to have the formation of what is called the platelet plug. So right now, a bunch of platelets are going to come in here and they're going to start plugging the cut on the blood vessel. Again, slowing down the loss of blood. Now, is this the end? No, it's not the end. Not yet. Then we come to step number 3. And step number 3 is the activation of different clotting factors. One of those clotting factors is what we call the tissue factor. And a tissue factor is a transmembrane protein that will lead to the activation, that combined with calcium is going to lead to the activation of an enzyme called thrombin. Again, tissue factor is going to combine with calcium, and that is going to lead to the activation of an enzyme called thrombin. What is thrombin going to do? Thrombin is going to take fibrinogen and transform it into fibrin. Fibrinogen is a soluble fiber. Fibrin is an insoluble fiber, creating this mesh that we're going to look at right here. Everybody look up. We are looking at a microscopic picture of a fibrin thread. And you can see that those threads are so tightly wound together that a red blood cell can even pass through it. It gets trapped in that thread. Okay? Now, again, what causes the formation of the fibrin thread? They are caused by tissue factor combining with the calcium to activate thrombin. Then, thrombin, with the help of vitamin K, is going to lead to the transformation of fibrinogen into fiber. Okay? So we need both vitamin K and we need calcium in order to be able to clot our blood. So if you are deficient in calcium, if you are deficient in vitamin K, you will bleed more because blood clotting is going to be compromised. Now, we have just seen, folks, how complex the process of hemostasis is, right? I have a question for you. Why do we need all of these steps in order to clot our blood? Think about it a second. Why not a one-step done blood clotted? What do you think? The answer is on the screen right here, by the way. Why does blood clotting need to be so complex? Let me ask you a different question then. Do we want to have intravascular clotting? You know what intra means, right? Intra means inside. Do we want to have clotting inside of a blood vessel without the blood vessel being damaged? I see everybody shaking their heads. No, we don't. Intravascular clotting is extremely, extremely dangerous. I'll talk more about that. Why? So the reason why the process of hemostasis is so complex is to make sure that intravascular clotting will not happen or decrease the chance of intravascular clotting. Because we only want to have clotting if there is damage to the blood vessel, which then is going to lead to a cascading reaction until the blood is clotted. But we don't want to be too easy to give the possibility of increasing the chance of blood clotting inside of a blood vessel. Okay? Now, nothing works perfectly in our body. We are not machines. So intravascular clotting can happen. And this is what we call thrombosis. So what is thrombosis? Thrombosis is the forming of a clot inside the blood vessel. And the clot itself also has a name. This is what we call a thrombus. So if we have a clot that is formed inside of a blood vessel, everybody look up here just for a second. Now we are looking at a blood vessel right here, okay? Little section of a blood vessel. And we can see that this blood vessel here has atherosclerotic plaque right there, right here. Now, what's going to happen when we have atherosclerotic plaque? What happens is that we're going to have the narrowing of the blood vessel, which means that the blood is going to slow down as it passes through that narrowing, right? If you have a smaller width, the blood is going to slow down. Every time that you have a slowing down of blood or blood that is pulling, that is sitting still inside of a blood vessel, that could lead to the formation of a thrombus, of a blood clotting. Okay, so atherosclerosis, trauma, infection, all of that can lead to the formation of a thrombus. Now, a thrombus is when the clot is attached to the wall of the blood vessel. Let's say there's a thrombus right here. If the thrombus detaches from the wall of the blood vessel and now starts to circulate in the bloodstream, now this is what we call an embolus. So an embolus is a thrombus that broke away from the vessel and now floats freely in the bloodstream. And this embolus, guys, can come all the way into the blood vessels of the brain, like we're seeing right here. And that can lead to what is called an embolic stroke. What is an embolic stroke? The lodging of an embolus in a small blood vessel in the brain, leading to the death of the brain tissue because the brain is not getting any blood to that section. We also have what is called a hemorrhagic stroke. What is a hemorrhagic stroke? A hemorrhagic stroke is when a blood vessel in the brain bursts, and now we have bleeding in the brain. So that's also going to lead to a stroke, but that is caused by a breaking of a blood vessel. This one here is caused by the lodging of an embolus. Any questions, guys? All right, moving on. Let's talk now, change gears here, and let's talk about the blood group type. Again, I have discussed that, started discussing that yesterday in the labs. And we do know that we have two different types of blood groups. We have the ABO blood group type and we have the RH blood group type. Okay, I'm going to talk about both of them. And both of these are relevant, are important when we talk about blood transfusion. Now, what makes a blood group type? What makes a blood group type is what we call the presence or absence of a surface marker, also called an antigen. So if you call it an antigen, if you call it a surface marker, it means the same thing. Let me give you an example. For the ABO blood group type, okay? The ABO blood group type is determined by the presence or absence of the A and B antigens on the surface of the red blood cell. If an individual has the A antigen, they're going to be type A, blood type. If an individual has the B antigen, they're going to be blood type B. If they have both A and B antigens, they're going to be type AB. And if they don't have the A or the B antigen, then they're going to be a type. It is as simple as that. That's what it means, the type A, B, AB. Okay? Now, what about the RH, though? RH is a different surface marker, is a different antigen. If you have the RH on the surface of the red blood cell, you are RH positive. If you don't have the RH on the surface of the red blood cell, you are RH negative. It is as simple as that. So you could be a type A positive or negative, a type B positive or negative, AB positive or negative, and even a type O positive or negative. Is that clear so far, folks? Now, When we talk about blood group type, not only we need to worry about the antigen, about the surface marker on the red blood cell, we also need to be concerned about the antibody that is circulating in the plasma of that individual. Okay? Because when you are a type A and you have the A antigen on the surface of the red blood cell, automatically, you're born this way, you will have antibodies against the B antigen, what we call the anti-B antibody. If you are a type B, you're going to have antibodies against the A antigen. If you are a type AB, you are not going to have antibodies against the A or the B antigen. And if you are a type O, you're going to have both antibodies against the A and the B antigen in your bloodstream. Please don't confuse antigen with antibody. It happens all the time. Even though I say don't confuse the two. Antigens are found stuck on the surface of the cell. Antibodies are proteins circulating in the bloodstream. They are not attached to the red blood cell. They're circulating freely in our plasma. Okay? And antibodies are highly specific to antigen. An antibody can only bind to one antigen. The anti-A antibody can only bind to the A antigen. The anti-B antibody can only bind to the B antigen. Okay? And once an antibody binds to an antigen, the antibody will destroy the red blood cell. This is why we need to be careful when having a transfusion. If I take a blood from a blood type B and I give it to an individual that is a type A, the antibodies in that individual's blood is going to find the antigen, the B antigen, is going to bind to it, and is going to literally chop up the cells in 1,000 little pieces and destroy it. Okay? And that is very dangerous. So, with that in mind, let's introduce the concept of a universal donor and a universal recipient. Again, specifically talking about the ABO blood group. And a universal donor is a type O. Why? Why can this individual here donate blood to anybody? The reason is they have no antigens on the surface of the red blood cell. So it doesn't matter what antibody you have, because an antibody needs an antigen to bind. So you can give blood to anybody without any worry about the red blood cells being destroyed, because there are no antigen on the surface. So which means that the type AB is the universal recipient of blood. This guy right here can get blood from anybody. Why? Because they have no antibodies against the antigens. So it doesn't matter what antigen you have. If I don't have antibodies to bind to it, they're not going to be destroyed. They're going to be left. So let's do a little blood typing right here together. So hopefully all of you do know your blood type. If you don't, fortunately, it's something that is very easy to determine. All that you need is untreated blood, a flask that contains anti-A antibody, a flask that contains anti-B antibody, and a flask that contains anti-RH antibody. And then you can type for both the ABO and the RH. Okay? All right, so when I tested this individual number 1 right here, when I added the anti-A serum, the anti-A antibody, we can totally see it that it reacted with the cells and destroyed the cells. When I added the anti-B serum, nothing happened. So I can tell right away that this individual is a type A, because the antibody found the A antigen, bound to it, and destroyed it. This destroyed the red blood cells. So individual 2 then is going to be a type B. Individual number 3 is going to be type AB because both antibodies reacted. And individual number 4 is going to be a type O because none of the antibodies reacted. Guys, I will ask you questions about this in the exam. And I can tell you from a previous experience that at least half of you will get these questions wrong in the exam. Even though I am telling you this will be on the exam. So my question to you is, do you have a question? Does this make sense? Is it clear? Why nobody have a question now, but half of you have a question on the exam? I can't help you on the exam. Can't help you now. It's a lost cause. Any questions, folks? So again, we do have to be very careful with blood transfusion. It can be deadly, guys. Receiving the wrong type of blood will lead to the clotting and the clumping of the red blood cells, and that can lead to an embolus. You can get a stroke and die. You can have a heart attack and die because it's going to clog the small blood vessels in the. Now, what about the RH though? The RH antigen is not as important, as crucial as the ABO when doing blood transfusion. Okay, it is still important, but not as important. Okay, now we know that you are Rh positive if you have the Rh antigen, or you are Rh negative if you don't have the. Let me tell you this: if you are Rh negative, that means you don't have antibodies against the Rh antigen. You've never been exposed to the antigen, so you don't have antibodies against. Let's say that you are a type A negative and you need a blood transfusion right away, otherwise you're going to die. And the only blood available, let's say it is a type A positive. Can you get blood from that individual? What do you think? Again, you are A negative. You need blood or you're going to die. The only blood available is an A positive. Can you receive that blood?
Yes.
Yes, you can. Yes, you can. Now, what is the issue then? The issue is you can only receive an Rh positive blood one time in your life. Because once you get that Rh positive blood, you will start developing antibodies against the Rh antigen. Let's say you need blood again a second time. If you receive blood the second time, if you receive blood from an Rh positive, now your antibodies are going to bind to the Rh antigen in that donated blood, and it's going to destroy the red blood cells, and that can be dangerous. So you can only receive it one time. Okay? But if you are Rh positive, it doesn't matter if you're receiving Rh positive or Rh. Yeah. Now, there is an issue with the Rh and the blood group type incompatibility with a mom and a fetus. Let's say that a mother is Rh and the fetus is Rh positive. In the womb, guys, there's going to be a mixture of blood between the fetus and the mother. Okay, so the mom is going to be exposed to the fetus blood, and because she's exposed to the fetus blood, the mom will develop antibodies against the Rh antigen. Now, this is not an issue for the first pregnancy because it takes time for enough antibody to develop, and by that time, the baby will be. So no issues with the first pregnancy. Let's say she gets pregnant the second time, And again, the fetus is Rh positive. Now, this will be an issue, not for the mother, but for the fetus. Why is it an issue? The issue now is that the mom is carrying our anti-Rh antibodies. And the antibody is going to find the Rh antigen on the red blood cells of the fetus and will destroy the red blood cells of the fetus, causing what we call hemolytic disease of the newborn. And this baby could die in the womb due to this hemolytic disease. Okay? Fortunately, there is a treatment for that, especially in developed countries where we have medical care, we have prenatal care, in which if the doctor knows that the mother is negative and the fetus is positive, and they're going to test for that, after the first pregnancy is done, right soon after, the doctor will inject the mom with a serum called RhoGAM. And what RhoGAM is going to do is going to prevent the formation of anti-Rh antibody. So if she gets pregnant the second time, she won't have the antibodies against the Rh antigen. So the second pregnancy is going to be just fine. All right, let's watch this video here on blood group type.
Take the next step in treating your moderate to severe ulcerative colitis with Entyvio, the only gut.
A blood type or blood group is a means of classifying blood based on the presence of specific antibodies and on whether red blood cells have specific inherited antigenic substances on their surfaces. There are 38 of these blood group systems based on various antigens. These antigens can be proteins, carbohydrates, glycoproteins, or glycolipids. The most important blood group systems are those that denote ABO and Rh status, since these determine suitability of blood for transfusion. We will be discussing them in this video. The ABO blood group system involves 2 antigens and 2 antibodies found in human blood, antigen A and antigen B, and antibody A and antibody B. The A and B antigens are found on red blood cells, while antibodies A and B are in the serum. The antibodies are usually IgM or immunoglobulin M antibodies. Humans have one of 4 combinations of antigens, along with a corresponding combination of antibodies, which we'll get to next. People with blood type A have antigen A, people with blood type B have antigen B, people with blood type AB have both antigen A and antigen B, and people with blood type O have neither antigen. If you're blood type A, then you have antibody B. If you are blood type B, you have antibody A. If you are blood type AB, you have neither antibody. Finally, if you are blood type O, you have both antibody A and antibody B. If antibody A encounters antigen A, or if antibody B encounters antigen B, an agglutination reaction results. This is very dangerous, and we'll cover this in a follow-up video. Blood transfusion is safe only as long as the recipient's serum doesn't have antibodies for the donor's blood cell antigens. The second most significant blood group system for blood transfusion is the Rh system. Rh stands for rhesus, and it involves 50 antigens. The most significant Rh antigen is the D antigen, as it's the most likely to cause the immune system to react. The presence of D antigen is signified by a plus sign after the ABO blood type. Lack of the D antigen is signified by a minus sign. For example, someone who has B antigen and Rh antigen would be signified as B positive. It's common for someone who is D negative to not have anti-D antibodies, but these people can develop these antibodies after a sensitization event. This can happen, for instance, during a blood transfusion, or during a fetal-maternal transfusion of blood during pregnancy. This can result in the development of Rh disease. So a D-negative patient without sensitization to D-positive red blood cells can receive a transfusion of D-positive blood once. Then they become sensitive to D antigen, so a future transfusion of D-positive blood would result in a dangerous transfusion reaction. And, in the case of D-negative females without prior sensitization, receiving D-positive blood results in a risk of hemolytic disease of the newborn. Hence, Rh D-positive blood is not given to D-negative women of childbearing age. Meanwhile, D-positive patients do not react to D-negative blood. The same kind of matching is done for other Rh antigens. Now let's apply what we've learned to fill out this red blood cell compatibility table. O-negative is the universal donor. Since there are no A, B, or D antigens, O-negative can donate blood to anyone without triggering an immune response from A, B, or anti-D antibodies. AB positive is the universal receiver, since there are no antibodies that will trigger an immune response to A, B, or D antigens. Blood plasma is the liquid component of blood which holds blood cells and other constituents of whole blood in suspension. This is where you find A, B, or anti-D antibodies. Hence, plasma compatibility is the inverse of red blood cell compatibility. Someone with blood type O can receive plasma from any blood type, while someone with blood type AB can only receive plasma from other people with blood type AB. Someone with blood type O can only donate plasma to others with blood type O, while those with AB blood can donate plasma to anyone. It must be noted that with rare exceptions, an individual has the same blood group for life. On rare occasions, it can change through addition or suppression of an antigen during infection, malignancy, autoimmune disease, or due to a bone marrow transplant. If someone with a different ABO type donates bone marrow to you, your blood type will eventually convert to the donor's type.
All right, guys, any questions? Briefly here, let's talk about some difference between the fetal hemoglobin and adult hemoglobin, in which they actually quite different one another. The hemoglobin of a fetus, folks, it has a greater affinity for oxygen gas than the hemoglobin of an adult. So HbA is an adult hemoglobin, HbF is a fetal hemoglobin. Why do you think that is the case? Let's think about this one for a second. Why do you think that the hemoglobin of a fetus needs to have a greater affinity, a greater capacity to bind to oxygen gas than the hemoglobin of an adult? Little critical thinking here. What do you think, guys?
Can you repeat that question?
Yeah, the question is, why does the hemoglobin of a fetus has a greater capacity to bind to oxygen gas than the hemoglobin of a... How does that, is that the case? Why did we evolve to be, how does it benefit fetus?
Is it maybe because the pregnant mom is breathing in the oxygen, and so the fetus has to share the oxygen going through her blood, and as a result, the fetus has to be more sensitive to pick up like oxygen because the mom is also using that oxygen too?
Absolutely. Absolutely. I don't know if how many of you guys had babies, but when you have a baby, you are growing a little parasite inside of you. Look, it's an adorable little parasite. Love my son, but they're still parasites when they are inside of the womb. They depend on the mother for everything. That is the definition of a parasite. Now, this little adorable parasite needs to compete with the mom for oxygen gas. And the only way that it can compete with the mom is by the fact that the hemoglobin of the fetus can grab on into that oxygen with more tightly, a greater affinity than the hemoglobin of. Okay? But as soon as that fetus is born, that greater affinity it goes. Hi, folks. So this is all for this chapter. We do have about 15 minutes. I do want to get started on chapter 11, just to make sure that we can finish both of this chapter this week. And you guys are going to have in fact two quizzes that's going to be due Sunday for chapter 10 and the chapter 11. And the chapter 11 is a continuation of chapter 10. We're going to talk about the cardiovascular system. Okay? Now, when we talk about the cardiovascular system, this is actually two systems in one. We have the cardio, which is in relation to the heart, and we have the vascular, which is in relation to the blood vessels. And they work in coordination. We need both of them in order for them to work properly. So we're going to talk everything about the heart. We're going to look at how does a heart beat. We're going to look at our pipes, our blood vessels, and we're going to finish this chapter talking about a very, very serious condition called high blood pressure, what we call a silent killer. We're going to explain to you why it's called a silent killer. So let's start from the basic here, okay? Where is our heart? This is not a tricky question. The heart is located within the thoracic cavity. It is actually found between ribs number 2 and ribs number 5. Now, I want everybody to look up here, and you're going to see a lie that has been told them. Everybody's been telling you since you were little. Our heart is not just located in the left side of the chest. I know that we put our hand over the left side when we're singing the national anthem. But if you can look right here, folks, we still have about a third of the heart that is actually on the right side of the chest. So yes, most of the heart is on the left, but it's still a third, 1/3 of the heart is on the right side. Okay? Our heart is sitting in a location within the thoracic cavity called the mediastinum. Again, I want you to understand the mediastinum is not a membrane. Mediastinum is a location within the thoracic cavity where the heart is found. And the heart sits right on top of the diaphragm, right there. And if you take a close look at the heart, we can see that our heart has a triangular shape to it. Okay? In which the base of the triangle, the base of the heart right here, is going to be pointing towards the right side of the body. This is the right. This is the left. While the apex of the triangle or the apex of the heart is going to be pointing towards the left side of the body. Why does this matter? Everybody pay attention here, please. It matters because several of the structures of the heart are bilateral. And you know what that means. Okay? And the way that we divide the heart in half is by passing an imaginary line from the base of the heart. The line is going to go through the intraventricular wall. We're going to talk more about it. And then the line is going to exit through the apex of it, right there. So this is the left side of the heart. This is the right side. We need that information. Okay? Now, our heart is made of 3 different layers. 3 different layers that make up the heart wall. We have an inner layer called the endocardium. Endo means inside. We have a middle layer called the myocardium. Myo means muscle. And then we have an outer layer called the epicardium. Epi means surrounding. Okay? So again, we have endocardium, myocardium, and epicardium. Guys, the epicardium is the same as the visceral pericardium. Remember, we talked about the fact that our heart is surrounded by a double-layered membrane, right? A membrane that is folded in half. And this membrane is called the pericardial membrane, but because it is double-layered, there's 2 sections to it. We have the visceral pericardium, and we have the parietal. So visceral layer, the parietal layer. And we can see how the epicardium is going to come in here, and then it's going to fold on itself, and then it's going to form the parietal layer on top right there. And in the middle, what we have, guys? We have a cavity. What is the name of the cavity? The pericardial cavity. Inside of the cavity, we have a fluid. What is the name of the fluid? The pericardial. Okay, and we can see, everybody look up here. We can literally see this pericardial membrane surrounding the heart. It's quite thick. as an enemy. Now, here we have a nice little model of the heart, and we can see all these coronary blood vessels, which we're going to talk more about later. But right now, what I want to do is this. I want to take this heart here and I want to slice it open. And I'm going to do a frontal or coronal section, just like that. And we can see that our heart has 4 chambers, 4 spaces. And each one of those spaces, each one of those chambers, they have a name. Before we name them, let's split the heart into tubes. Again, how we do that? We're going to pass an imaginary line from the base of the heart, going through the interventricular wall, the wall separating the ventricles, exiting through the apex. And here we have left side of the heart, right side of it. So now we can name the chambers. And we have 2 atria and 2 ventricles. We have the right atrium, left atrium, right ventricle, left. And I will ask you guys questions about this in our next exam, next week, Thursday. You need to know the name of the chambers of the heart. Okay? Now, these chambers here are attached by major blood vessels that we call the great walls, the great vessels of the heart. And we're going to talk about some of these great vessels. Now, what is the function of the right atrium, folks? The right atrium receives all the deoxygenated blood coming from the body. The deoxygenated blood coming from the neck and the head is going to arrive in the right atrium through what we call the superior vena cava, which is a great vessel of the heart. The deoxygenated blood from the rest, the lower part of our body, is going to arrive in the right atrium through the inferior vena cava. So now the blood is inside the right atrium. So the right atrium is filled with blood. Now the right atrium is going to squeeze, is going to contract. We're going to talk more about that later. And now the blood is going to be pushed inside the right ventricle. Now the ventricles will contract. They're going to squeeze, and they're going to push blood out of the heart. From the right ventricle, the blood is going to enter what we call the pulmonary trunk, right here. And the pulmonary trunk is another great vessel of the heart. And this pulmonary trunk is going to split into 2 branches. We have the right pulmonary artery, and we have the left pulmonary artery. Okay? So we have left pulmonary artery, we have right pulmonary artery. Where is this pulmonary artery is going to go? The pulmonary artery is going to take the deoxygenated blood and carry that all the way to our lungs. What is the blood is going to do in the lungs? The blood is going to become oxygenated again. We're going to dump the CO2 into the lungs and grab the oxygen gas. And now this blood, this oxygenated blood, needs to return to the heart. It's going to return to the heart through what we call the pulmonary veins. And again, these are bilateral. We have the left pulmonary veins and we have the right pulmonary. Now the question is, why do we call one a vein, the other one an artery? Guys, arteries take blood away from the heart. This is why this one here is called a pulmonary artery, even though it is carrying deoxygenated blood. Now, these ones right here are called pulmonary veins because they are returning blood to the heart. Okay, so arteries take away from the heart, veins bring it back to the heart. Okay, now all the oxygenated blood is going to arrive inside of the left atrium. From the left atrium is going to go into the left ventricle, and from the left ventricle Now the blood is going to enter what we call the ascending aorta. The aorta is divided into different sections, and you need to be specific when you're labeling the aorta. It is not enough just to say aorta. The aorta is divided into ascending aorta, in which the blood is moving up. We have the aortic arc, or the arc of the aorta, and we have the descending aorta all the way down. Okay, so ascending aorta, aortic arc, and descending. This is going to send the blood to the rest. Any questions? Look, you do need to know all of this for our next exam. Now, we do have a different circulation of blood. We don't have just one type. We have several different types. I'm going to talk about 3 of them right now. We have what we call systemic circulation, we have a pulmonary circulation, and we have a coronary circulation. What is systemic circulation? Systemic circulation is the circulation of blood between the heart and the entire body. That is systemic. Pulmonary circulation is circulation of blood between the heart and the lungs. And a coronary circulation is a circulation of blood to the walls of the heart. Okay? Going to the walls of the heart. Now, in the systemic circulation, the arteries are carrying oxygen-rich blood, and the veins are carrying oxygen-poor blood. But look at this, guys. In the pulmonary circulation, that is the opposite. In the pulmonary circulation, the arteries are carrying deoxygenated blood, and the veins are carrying oxygen-rich blood. So this is why we cannot say that an artery always carries oxygenated blood, because it is not always true. It is true for the systemic circulation, not true the pulmonary circulation. Okay. Now, let's take a look at this heart right here that I sliced open. Everybody look up, please. And I circled right there the left ventricle. Okay, so this is the left ventricle right here. This would be the right vein. And I want you guys to notice, I mean, you can't miss it. Look how thick the wall of the left ventricle is in comparison with the walls of the right ventricle. Can anybody tell me why? And come on, this is the last time I'm going to bother you today. Why is the left ventricle thicker? The walls are thicker than the right ventricle.
Because there has to be like a higher pressure to get the blood out, or there's like to get the blood out to the whole body.
Yes, say it like a minute, and absolutely. Remember, from the left ventricle, the blood is going to go into the aorta and going to be distributed all over the body, all the way to the cells of my toe. So the amount of pressure that we need to squeeze the blood out is greater. So this is why the walls are thicker, more muscular. Now, for the right ventricle, the blood is only going to the lungs. From the right ventricle, it's going to go to the lungs to become oxygenated. And the distance is very short. This is why the walls are thin. All right, guys, any questions? Okay. Important. The quiz for chapter 10 is open right now. Okay, you will have until Sunday night to take it, but I would advise that you take it ASAP, because you're going to have 2 quizzes due this Sunday. The quiz for chapter 10, which is open now, and the quiz for chapter 11, which I'm going to open on Thursday. Okay? Have a great rest of your day. I'll see you guys next. Thank you.